Welcome students to exercise 30, which is going to focus on the anatomy of the heart. We will actually be discussing the cardiovascular system for quite some time here in lab. Blood pressure, EKGs, blood vessels, all of that stuff. But we're going to start really with the structure of the heart. Starting with where the heart is actually located in the body. And it's right here in the thoracic cavity, actually within kind of the middle portion of the thoracic cavity called the mediastinum. The base is the superior portion of the heart. The apex is the inferior portion of the heart, and it points off to the left hip. If we look at the kind of wall of the heart, and we look at the kind of layers within it, and as well as the kind of pericardial sac that it sits in. The most superficial here is the fibrous pericardium, so this really strong kind of fibrous collagenous tissue that surrounds the heart and protects it. And then we get into the two layers of the serous membrane. So we have the outer parietal layer separated from the inner visceral layer, which is also known as the epicardium, by the little pericardial cavity here, which then fills with fluid. It's a nice kind of friction-reducing structure, the serous pericardium. Then we actually get to the muscle layers of the heart, the myocardium. Myo means muscle. The heart is cardiac muscle, and cardiac muscle exclusively. You'll get a chance to look at that, what it looks like microscopically. And then we get the endocardium, which then lines all of the chambers of our heart. In terms of the chambers of our heart, we have 4 of them, like some other animals like birds. So all mammals have 4-chambered hearts. We have 2 atria and 2 ventricles. The ventricles are much larger than the atria. The ventricles are the pumping chambers, so they need more muscle. The atria are the receiving chambers, so they get the blood from the body. The right atrium receives the oxygen-poor blood from all of our body tissues via the inferior and superior vena cava. So the superior vena cava drains all of the kind of upper body regions into the heart. The inferior vena cava drains all of the lower body regions into the heart. The left atrium receives oxygen-rich blood from the pulmonary circuit, from the lungs, via these right and left pulmonary veins. If we look at the 2 ventricles, the right ventricle pumps the oxygen-poor blood to the lungs via the pulmonary trunk here, which very quickly branches into the left and right pulmonary arteries. And then the left ventricle pumps the oxygen-rich blood out to all of our body tissues via the aorta. So since we have 2 chambers on each side of the heart, right and left, 2 sides of the heart, we essentially have 2 adjacent pumps. The heart is considered a double pump system. The 2 atria are separated by the inner atrial septum, a little piece of kind of fibrous tissue that kind of separates them. The 2 ventricles are separated by the inner ventricular septum. And you can actually get holes in the septa, both interatrially and interventricularly, and kind of depending on the size of the holes, it can kind of interfere with your heart function. So the right side of the body gets the oxygen-poor blood from all of our tissues. It then pumps the blood to the lungs in order to get rid of the carbon dioxide and to pick up oxygen. So the right side handles what we call the pulmonary circuit. The left side receives all the oxygen-rich blood from the lungs and then pumps it out to all of our body tissues via what is known as the systemic circuit. And so if we look at that, you can see 2 pumps, double pump. The right side of the heart receives the blood from the body and then pumps it to the lungs, where it becomes oxygenated. It then travels back to the left side of the heart, where then the left side of the heart pumps it out to the body. Our body uses all of the oxygen, and the oxygen-poor blood goes back to the heart, lungs, heart, body, etc., 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 all the live long day. Never stopping. So since we have multiple chambers here, and some very large blood vessels. We have to make sure that blood is only flowing in one direction. We never really want it to go backwards. And to help us do that, we have two sets of valves, 4 valves altogether. We have 2 atrioventricular valves, or AV valves, and 2 semilunar, or SL valves. The 2 AV valves are the mitral or bicuspid valve on the left side, and the tricuspid valve on the right side. The AV valves will be open during diastole, or relaxation of the heart, and then when the heart begins to contract, that kind of forces shut the AV valves so that blood doesn't flow from the ventricles back into the atria. The semilunar valves are the aortic valve, which separates the left ventricle from the aorta, and the pulmonary valve, which separates the right ventricle from the pulmonary trunk. These valves will open during systole, or contraction of the heart that allows blood to be ejected from the heart into the big blood vessels, and then will close as the heart relaxes in diastole so that blood doesn't kind of flow backwards back into the heart. We don't want it to do that. All 3 of the heart valves, or all 4 of the heart valves with the exception of the left AV valve, have 3 what we call cusps, or little cups. They're basically little flaps of tissue. The mitral or bicuspid valve is the only one of the valves with 2, hence bicuspid. If we look at the cardiac circulation, what we are talking about are the blood vessels that supply and drain the heart itself. So the heart is an organ, the heart is a muscle. It is constantly pumping and pumping and pumping and pumping and pumping, and so it requires a very large oxygen supply. It's not that the blood inside the chambers kind of like diffuses out into the heart muscle. The heart itself has to be supplied and drained of blood. And it's the cardiac circulation that does that. So if we look at the 2 big arteries, what we really have a branch off of the ascending aorta is the left coronary artery and the right coronary artery. The right coronary artery then branches into the right marginal artery, and then on the back side of the heart, the posterior interventricular artery. The left coronary artery branches into the circumflex artery, which then goes around the rest of the heart, and the anterior interventricular artery. And that's how the blood gets out to all of the heart muscle itself. Draining the kind of now used deoxygenated, oxygen poor blood from the heart are several cardiac veins. On the front of the heart, we've got the great cardiac vein, small cardiac vein, and on the back of the heart, we have the middle cardiac vein, the great cardiac vein, the small cardiac vein, and the middle cardiac vein drain into this big kind of flat blood vessel called the coronary sinus. And the coronary sinus actually dumps its blood into the right atrium so it can go be pumped to lungs and be oxygenated. We also have these anterior cardiac veins, and they dump directly into the right atrium, so they don't go to the coronary sinus. But that is how the heart gets its own supply of blood and is also drained then of its supply of blood. One of the things that we want to do is look at cardiac muscle kind of both macroscopically and microscopically. If we look at the gross anatomy of the heart, you'll see that the heart tends to kind of form in these kind of spiral-shaped bundles. This makes a very specific pathway for electrical signals to travel, and that's important in terms of the functionality of the heart, which we'll talk about a little bit later in the semester. We also will look at cardiac muscle kind of microscopically. It is a striated muscle, so it has sarcomeres. It is not kind of straight. It's very much a branching type muscle. The cardiac cells themselves tend to be kind of short and fat, and then they are connected by these very special connections called intercalated discs. And the intercalated discs have kind of a twofold purpose. They have two things in them. They have desmosomes, which act like Velcro, and they have gap junctions. So the desmosomes prevent the cardiac muscle cells from separating as the heart pumps, and the gap junctions allow ions from one cardiac muscle cell to very quickly move to another cardiac muscle cell, so that the cells can transmit information from one cell to the next very easily. It allows the heart to function as a unit. We don't want kind of like one part of the heart to contract and the other part of the heart to contract. We kind of want it to do it all at once. And the gap junctions allow us to do that. And then the desmosomes allow the heart muscle cells to stay together despite constant stress of the kind of constant pumping action and contraction. So key feature in terms of cardiac muscle, intercalated discs. One of the best ways to understand the structure of the heart is to understand how blood flows through the heart. So from the systemic circuit, through the right side of the heart, through the pulmonary circuit to the lungs, and then back to the heart and out to the body. And so we'll start really with where blood comes from as it moves into the heart. So we're getting from the upper body, from the lower body, and then from the coronary sinus, from the heart itself. So the vena cava and the coronary sinus bring blood from the body and from the heart into the right atrium. This blood is oxygen poor, deoxygenated blood. The right atrium is then separated from the right ventricle by the tricuspid valve. So blood has to pass through the tricuspid valve. If you think of the valve as a door into the right ventricle, the right ventricle then contracts. Blood moves past the pulmonary semilunar valve, another door, into the pulmonary trunk. The pulmonary trunk then branches into the pulmonary arteries, and it is those pulmonary arteries that brings the blood to the lungs, where it can then percolate through all of the capillaries of the lungs and pick up oxygen. This now oxygen-rich blood returns to the heart via the pulmonary veins into the left atrium. From the left atrium, the blood moves past the mitral valve into the left ventricle, and then the left ventricle contracts, the aortic semilunar valve opens, and blood is pumped from the left ventricle into the aorta and out to all of our body tissues. And so if you understand that, if you understand how blood flows through the right side of the heart to the lungs, back to the heart, and so on and so forth, you have a really good grasp on what the heart looks like, or structurally, how the heart is structured, what vessels connect to what vessels, what valves separate which chambers, how the chambers work. So understanding blood flow through the heart is a really good kind of synthesis question in terms of understanding heart anatomy. One of the other things that you guys are going to get a chance to do in lab is a sheep heart dissection. So we are mammals, sheep are mammals, and so the sheep heart is a really good kind of stand-in for a human heart. So you'll have to make sure that you do wear closed-toed shoes to lab that day. You have to be careful. You have to read carefully. You have to pay attention. Don't be messing around with the dissection tools. You can use the probes here to help you identify the different blood vessels. And really what you want to do is essentially take your time, and all of the structures that are on our little heart anatomy list, you want to be able to identify on the sheep heart. Because on your lab practical, we're going to put sheep hearts out again with little pins stuck in them saying, what is this thing, what is this thing, what is this thing? And so you want to be able to identify the different chambers, the different valves, all of the blood vessels at the base of the heart, etc., etc., etc. And that's the end of our little lecture here. Looking forward to seeing you guys for our sheep heart dissection.